Hello everyone, my name is Deepti Varshney. I work with European Galaxy team in Freiburg and my research focuses on plant genomics and related area. Today in this video, we will be focusing on identification and evolutionary analysis of transcription-associated proteins in streptophyte algae and land plants. In this tutorial, our main objectives would be to understand the role of TAPs. We will learn about how to identify the TAPs from the given proteome using TAPscan tool, and we will be learning about how can we construct the phylogenetic tree for the identified TAPs. But before jumping into the analysis section, let's just understand what TAPs are. So the regulated expression of the genes is essential for defining morphology, functional capacity and developmental fate of both solitary living cells as well as the cell inhabiting the social environment of the multicellular organism. In this regard, the regulation of transcription, that is the synthesis of messenger RNA from the genomic DNA template, play a crucial role, as it contributes to control the temporal and the spatial RNA and the protein levels in the cell, and therefore has an essential function in all the living organisms. And in this regard, TAPs came into the section. So TAPs are the essential players in the gene regulatory networks as they involved in transcription regulation. These proteins are broadly classified into transcription factors and the transcription regulators. Transcription factors bind sequence specifically to regulatory elements that can enhance or repress the transcription process. On the other hand, TRs are involved in protein-protein interactions, or you can say that these proteins serve as co-activators or co-repressor. Additionally, there are some proteins that refer to as a putative TAPs that are thought to be involved in regulation of transcription, but their exact function is undefined. So, in the order of identifying these proteins, we will be using in this tutorial TAPscan V4 tool. This is a comprehensive tool for annotating TAPs with a special focus on species belonging to the Archaeoplastica. In general, the detection of TAPs is based on the detection of highly conserved protein domains. So in this tutorial, we will be learning about identification of TAPs in streptophytes algae and land plants using this TAPscan classifier, followed by the construction of phylogenetic tree. So now let's just get started. So to get started in this tutorial, we will be using the representative protein sequences obtained from the Genome Zoo database. The selected sequence represent a different lineages. So we will be using a sequence from streptophytes algae, bryophytes and the land plants, and first we will be identified the transcription associated proteins in all of these species, and then we will choose one of the gene family from the output and then perform the phylogenetic tree analysis for them. Okay, so now let's just get started. So first thing we will be doing is create a new history. So we will create a new history for this tutorial and give a proper name. Okay, so let's create new history. Like if you click on this plus icon, it will allow you to create new history. So first we will give a name to this history. So for me, I'm giving a name, it's TAP genes. You can give any other names. Okay, so TAP genes. After creating a history and giving a proper name, you can import the data. So I will be using the data which is already available on our server, in the remote server. So for that I will be using the data from the Zenodo link. So for that I will just copy these links and go back to my tutorial, click on this upload section. I will click on paste and fetch because I have copied the links, and just paste all the links here, and then click on start. As soon as you will start the process, you will be seeing all the files are appeared in my history. So we will just close this one, and yeah, this will take some seconds to get it uploaded. So if you see this gray color, that means your job is in the queue. So, and now you see it turned orange, so that means your job is running. So now you see all the files have been uploaded, turn into green, so that means our file is already on our Galaxy history. Now to give you a very quick overview about these species FASTA here. So we are using 2 species from the streptophyte algae and the 5 species from the bryophytes and the land plants. And to have the more information, like we are just using the FASTA file with these 5-letter codes. So 5-letter code .fasta, just to have a small name for these files. So we have this. So CHABR, that belongs to the Cara brownie, and thus PENMA, it belongs to the Penium margitaria. So just to remember that okay, this file belongs to algae, this file belongs to bryophytes. Okay, so as now we have this file in our history, we will create a dataset list, because we want to analyze all these files all together in order to identify the tabs. So we'll click on this checkbox, click on this select all, and then there is a drop down option. We have to click on this and click on build dataset list. So you'll see all the files are here now, and we will give a proper name to this list. We can say input sequences, yeah, and then create a list. So now it will create a list for us. So yeah, this is a list in which we have these FASTA. It will just loading 1 2 3 4 5 6 7. So we have 7 files here. So we have created history, we actually gave a name to this history, we have imported the input sequences via Zenodo link, and also created a dataset list. And now we will identify the tab sequences. So in order to detect the tabs, first each sequence from our species FASTA will be first scanned for the protein domains. And based on the domains, TAPscan classify applies the specified rules to assign the protein sequence, and it's scanned for 137 different families to ensure accurate family assignment using the GA threshold and coverage value. So in short, first our sequences will be scanned to identify the protein domains, and based on these domains, the TAPscan will assign the tab families for the sequences. So first we have to click on this TAPscan tool. We need to provide this dataset collections which we just created as an input. If you want to have the domain output from the HMM scan from the hammer analysis, you can click on yes, and then just click on run tool. Okay, so while this TAPscan running, we will understand what kind of outputs we can expect from the TAPscan. So yeah, so TAPscan classifier provide us with 3 different outputs. and all of these outputs will be in the tabular format. So in output 1, we will be getting the information about the TAPs that contain the detected domains, and finally assign the TAP family for each gene ID. And if domains are assigned to a sequence but not all the rules are fulfilled, the sequence is assigned to a no family found. I'll show you how it works. So in output 2, this will be the summary of the number of members for each TAP family. In output 3, we will be getting the information about subfamily. If there is any TAP which has divided into subfamilies, then in extra column we will be getting this information. So below is the example of detected TAP extra. So these kind of output we can expect in output 1 and output 3, but in output 1 you will be getting only 4 columns. One is sequence ID, TAP family, number of classification and domains. But in output 3 we will be having an extra column of subfamily here. So let's check the output here. So first we will be clicking on output number 1, so detected TAPs. So if you click on this, you will see all the outputs here. So we'll check for Arabidopsis thaliana. So as I told you that we will get the output in tabular form. This is our tabular output. So in the first column we will be having our sequence ID. The second column gives you the information about the identified TAP families. Third column of the number of classification for this protein, and then how many of the domains are identified in this particular sequence. Yes. So now let's take a look on the second output. The second output will give you the summary of the TAP family and the number of detected proteins in that TAP family. So as you can see, for example, 88 TAPs, there are only 2 proteins were identified. So, and you are seeing that no family found. So as I just told you that this happens when domain was assigned to the particular sequence, but at the time of scanning these domains using the TAP scan script, it doesn't follow or it doesn't pass the threshold for assigning the TAPs, and that's how it will be classified as a no family found. And in this Arabidopsis thaliana proteome, we have 675 proteins which were classified as a no family found. Now let's take a look on output 3 for the same species. So in output 3, as I told you that we will be getting one extra column, but all the other columns will be the same. So like sequence ID in the first column, TAP family name in the second column. Third column will tell you about the subfamily. If the TAP is divided into subfamily, then yes, and the number of classification in fourth column, and the domain, all the domains which is identified in particular sequences. So now, as we already identified the TAPs, now we will prepare our data to get the sequences for those identified TAPs and then perform the phylogeny. But first we have to decide about the TAP family, like in which TAP family we will be running this phylogenetic tree analysis. So for that, I will be using AUX/IA TAP family. This TAP is classified in the transcription repressor, and this helps in inhibit the expression of auxin responsive gene by dimerizing with auxin response factor transcription activators. So we will be using this TAP family and extract the sequences for this family from all the species and run the phylogeny. Okay, so in order to just filter the TAPscan output based on the column, we will click on this tool, and then because we have output and TAPscan selections, for that we can either choose output 1 or output 3, because output 3 and output 1 is more or less similar, but with just extra column in output 3. And because we have to filter our TAPscan output based on the second column in which we have the TAP family information, and as I said, we will be using this TAP family for our further analysis, so we have to put a filter for that. So we can just replace whatever it was here in default. So you can just select the detected TAPs with the following condition. Would be the second column should be AUX/IAA, and the number of header lines to skip, because we don't want to touch the header, so we will just say to skip the header line 1 and click Run Tool. So you see our filter tool has been finished. We'll see what we have now. You see all the lines which has TAP family auxin as a TAP family in the second column is just filter all these, so you can just examine the another file also. So in this case we only have 2 proteins which has TAP family here, so like this. Now after getting the filtered output, you see we still have all these 4 columns. We just want to work with one column, and which would be our sequence ID that can be used to extract the protein sequence later from the FASTA or from our main input file. So what we will do to get the first column, so we will be using this tool. It says cut column from table, so these are the text manipulation tools. So for this, as I said, that we will be working with only first column, so C1, and then you see it automatically appears. So filter, so we will be running this cut tool on filter output and then Run Tool. Okay, so these jobs have been finished. Okay, so this job is also finished, so we will examine the output now. So we ask from this tool to cut the column 1 only. So yeah, now you can see in the output we only have first column in which we have the sequence ID, but in the header line you see the sequence ID is still there. If we would like to extract the sequences for all these IDs, we just need to remove the first header line also from this. So for this, what we will do, we will use the another text manipulation tool which is called remove beginning of the file. See, so for that we will use the output from the cut tool and ask for the remove first, so which is our header line, and just click on Run Tool. This will remove our header line in which we have this sequence ID written. Yes, so right now it's running. Okay, so now let's check the output. Okay, So you remember earlier we had this sequence ID written in our first line, but now it's gone. So now we can actually use this output to extract the sequences. So for that we will be using this tool, filter faster, and see. So we'll click on this tool, select the database collections, and I hope you remember what we had in our first input. So we had the list. In the list we had multiple FASTA files. Just to give you a small overview about how FASTA file look like, if you don't know, then so we have this FASTA file for which starts from this sign, and then the first line gives you the information about the header, and the second line gives you the sequence. So okay, we will use this tool. So first we need to provide the FASTA sequences from which we would like to extract the sequences for our desired IDs. And then so we select this input sequence. Now we have to fill the criteria for filtering on header. So as we have the list of IDs from the remove beginning tool, and then we can just collect on datasets collection, and your most recent output will be appear on this automatically. So yes, so we will be using this as an input, or you can say for list of IDs to extract the sequence for, and click on run tool. Filter FASTA is also finished. Now let's just check the output from filter FASTA. So we'll click on this, we'll check. So here you can get the information about how many sequences we have extracted for Arabidopsis thaliana for this TAP family. So there are 29 sequences. Now we can just quickly check, do we have 29 TAPs for this TAP family? We can just quickly check, we just need to be sure that okay, we have the exact number of the TAPs extracted from the FASTA. So you see we have 29 lines. That means we had actually 29 sequences for that particular TAP family. So yeah, this is kind of one way to examine that okay, we are going in a right direction. You remember for the Kara also we had 2. So now we can just quickly check the another output from the filter FASTA and check. Let's see, we do have actually 2 sequences extracted. So you can see from this options, yeah, the 2 sequences, found 2, wanted 2, and then we had 2. So these are the sequences for the Kara brownie. Okay, so we have now identified all the sequences belonging to this TAP family across all the multiple species, and now we will be performing the evolutionary analysis on this data. So for that, first we need to align all these sequences by using the multiple sequence aligner tool. In this tutorial we will be using MAFT tool. So we'll click on this MAFT, and for multiple input generates, so as for now we have multiple FASTAs, right, and we would like to generate a single MSA output file of all the sequences from all the inputs. Yes, and in selecting the input batch we will have to click on this input batch, where we can provide our output, or you can say the output from the filter FASTA, where we have the FASTA sequences from this AUX TAP family. And now let's check for the other parameters. The type of sequences, we have to select the amino acid. instead of auto detect, we will just select amino acid. This will give us the surety that we are going to use only the amino acid for MAFT. Type of scoring matrix Blossom 62. We will click on this. This says the support unusual character, and in selecting of MAFT flavor we will just click on auto. Let's see if we did everything correct. So MAFT flavor auto, and then now we have to select the reorder output. So we will just check on yes now, and then click on run tool. So now you can see we will have only the one output file in which we will see the alignment. Our MAFT analysis also finished. Now let's just examine the output. So for examine the output, we have to click on this I button. It will show you the information about the output. So see, we have the output in FASTA format, but you can also see there are some other information like these dashes. So these dashes actually indicate the gaps in the alignment. So, and now we would like to visualize this output, because now you can see that this FASTA format output is not very informative to us, right? So to visualize this output in a graphical way, what we will do is we just need to click on this visualize icon here and click on this, and this will give you the inbuilt Galaxy viewer to check the output. Okay, here you can see, yeah. So now you can see this output, or you can see the output alignment output files. So if you click on the very right on this, if you can just drag it on the top right, you can see the alignment length for our sequences. So alignment length is around 1856 or 7. So we are at the end of the sequence here. So this is the output which we got from the MAFT. Okay, but as you can see, there are so many gaps we are seeing in this outputs, which doesn't look nice, right? So now what we will do is we need to clean this output file a bit. So in order to clean the output, what we can do is we can just run the trim al, which will remove, or you can see the clean up this alignment by removing the poorly aligned region from the output from the MAFT. Okay, so we'll click on this tool. We need to select the input file from our history, which would be our MAFT output, right? And we have to select the criteria on which we want to remove the poorly aligned region. So we will use the custom mode in the section of select trimming mode from the list. And for the other parameters, we have to assign gap threshold and the similarity threshold. So for the gap threshold, we will use 0.5, that means the 50% of the gaps threshold. And for the similarity, we will be using 0.001. So, and rest of the parameters will be will be as it is, and then we can just click on run tool. So what TrimAL does is it will remove all the poorly aligned region based on the threshold criteria which we just assigned. What kind of output we can expect from the TL? So it will give you the alignment, of course, but it would be the trimmed alignment, like we just assigned 2 parameters that we would like to remove the sequences, the poorly aligned regions which have the gap lower than 50%, and also the similarity less than 0.001. Okay, so seems like TrimAL job has been finished. So let's check the result. So likewise, we'll click on this I button again to check the output. So I'm just examining the HTML report right now. So here you can see the information about the filtered, or you can see the trimmed output. So we have selected sequence 85. You remember in the MAFT we had actually 85 sequences altogether, so that means none of the sequences actually removed. But if you see the selected residue, we have around 929, and the deleted residue, that means 928. So that means half of the residues has been deleted because of the poor quality by using TrimAL. And likewise we have the trimmed alignment file. This is actually very similar like the MAFT, but now you will be seeing less gaps in compared to MAFT output. See, here you can examine like this. We are going to use this output to generate the tree, and for this we will be using quick tree for the tree generation. So in the parameter we will be using the alignment file, right? So it's already selected, and as an output we will gonna use the output from the TrimAL, right? So single output, because now we have only one file, and we can just select the trimmed alignment output. And we need to mention about the bootstrap value with number of iteration, so we can just use the bootstrap value here. Okay, maybe for it's just loading, so 100, and then click run tool. So it will generate the phylogenetic tree by using the alignment information. So quick tree job has been finished. Now let's just take a look how our tree looked like. So as mentioned in the in the tutorial, the output from the quick tree will be something like this. So we have this information like this, but this is also not very informative, so we are going to use the inbuilt function of the galaxy, and which we can visualize the phylogenetic tree. So we just need to click on the visualize, and you can see the tree is here. Okay, so you can see this tree like this. Next, if you want to visualize this tree in a different manner, so like you want to add your alignment information into the tree, for that purpose you can use the ETE tree viewer tool, so for the tree visualization. So what we will do is we will use this ETE tree viewer, and we will provide our tree from the quick tree output. Let's see. So yeah, we need to provide the new week tree to visualize, and we can add the alignment information into the image. So for that we need to click on yes for add alignment information, and for the FASTA file, because we need to provide the output from the alignment, so we are going to use the clean alignment output which we got from the trimmed AL. So I have selected the trim alignment for the alignment FASTA, and now we need to check for the other parameters. So resolve taxonomic ID, so we can click on yes, so it says already yes. And format for the output image, either we can ask for SVG and the PNG. We are going to use PNG for us, so and click on run tool. So now the job has been finished for ETE3. We can visualize it by clicking on this I button, and you'll see the tree is still loading for us. Okay, so you can see we have the phylogenetic tree now, and we also have the alignment information here, so we can also display the alignment So ETE3 actually did this for us. And we also have the NCBI taxonomic information for each of the sequence related to these species, right? So what do we see in this tree first? If you look closely, you will see that all the species, or you can say all the sequences, belongs to the biophytes, and the land plants are clustering together. So you can see Oryza sativa, Arabidopsis, and in case of other species, Singela, Fiscometrium, these are actually clustering together. First thing, we only have very few AUX TAP families in the streptophytes algae, and in case of land plants and the bryophytes, we have more number of the TAP family. So this actually signifies that it could be possible that this family, this protein, the protein sequences from this family actually expanded as plant transitioned from the aquatic to terrestrial environment, where more complex signaling mechanisms were required for growth regulation. And also the phylogenetic tree analysis also support this finding that okay, the genes comes from the same group, such as the land plants and the bryophytes clustered together, indicating their close evolutionary relationship and potential for similar roles in auxin signaling pathway. So yeah, that was about the identification and evolutionary analysis of transcription-associated proteins in streptophyte algae and algae. So if you have all the outputs in green color in your history, that means you have already finished this tutorial. Congratulations. Thank you.